In addition to plasmid vectors and viral vectors, there are two other methods of delivering DNA into a living cell, and those are called electroporation and protoplasm fusion. During electroporation, small pieces of DNA can be delivered into a cell using electricity. The target cells are placed in culture with fragments of DNA around them. And then an electrical pulse is briefly delivered to the culture. What the pulse of electricity does is open up pores in the membrane of the cell. And that will allow the fragments of DNA in the environment to be taken up into the cell. In the drawing at the bottom of the slide, you can see a representation of a cell with a pink membrane around it. And then they're showing us very small fragments of DNA shown in green. The plus and minus charges are there to remind us that there is a normal sort of charge distribution inside and outside a living cell at all times. Now we're going to deliver an electrical pulse to the cell, and what will happen is that distribution of charge, all the pluses and minuses, will change, and that will open up pores in the membrane and allow the little green pieces of DNA to travel into that cell. Protoplast fusion is a little bit different. We haven't yet introduced this term protoplast. It's defined as a bacterial cell that has had its cell wall removed. So we have created in a protoplast a more typical cell where its membrane is exposed on the outside. Protoplast fusion is a process where two or more of these protoplasts are treated not with electricity, but with chemicals. And the chemical exposure will alter the cell membrane and allow them to fuse together. When the membranes of two or more of these protoplasts fuse together, it creates one big cell. So the cytoplasm will all get mixed together, and all of the DNA will get mixed together as well. The drawing down at the bottom of the slide shows two bacterial cells over here on the left. You can see one of them has a purple piece of DNA and the other one has a green piece of DNA. We're going to create protoplasts out of these bacterial cells by removing their cell wall. And that's what you're looking at here. Then we will treat these protoplasts in culture with the chemicals that will alter their cell membranes and allow those membranes to fuse. So now, instead of having two individual protoplasts, we have one big one. And all of the cytoplasm and all of the DNA is now inside this one big membrane-bound structure. What will happen is that this DNA that is now combined in here will naturally go through a process of recombination. And what will result then is the production of two new protoplasts that no longer have purple in one and green in the other, but now a combination of purple and green in both. The cells can then be allowed to rebuild their cell walls and then continue to grow and develop in culture. And we have created recombinant, genetically modified cells. Once a bacterial cell has been genetically modified through any of the processes that we've discussed so far, that cell can then be cloned. Remember, bacteria undergo cell division through the process of binary fission. And binary fission creates clones. It creates copies of bacterial cells that are genetically identical to each other. So once we have modified the DNA of one bacterial cell, by allowing it to grow in the laboratory, we're going to create lots of cells that have that same genetic modification inside them. Once we have lots of cells that have that new DNA, that genetically modified DNA, we can grow those cells and allow them to express that DNA. When they express the DNA, we're going to get protein made. And we can harvest that protein out of the culture and use it for our own purposes. Remember, sometimes what we're growing this way is pharmaceutical product. And sometimes we're growing other types of proteins, enzymes and things like that, and other types of proteins that are useful commercially. The other thing that we can do once we've modified bacteria is we can recover more copies of the gene. In other words, the more cells we have that contain the modified DNA, the more copies of the gene are in that culture. And we can now harvest all of these copies of this gene out of this culture and use them for other purposes. We might take those copies of the gene and insert them into other organisms, for example. So yes, we do use bacteria quite a bit in biotechnology, but we also use other cell types, yeast, as I've mentioned, particularly the type of yeast called Saccharomyces cerevisiae. This particular type of yeast is very good at taking up plasmids from the environment, and it's also very good at secreting protein products. In other words, there are certain genes that code for proteins that these yeast cells will secrete into the culture, and those are easier to harvest for us. Of course, there are also plants that undergo genetic modification. As I mentioned before, plants like corn and soy, even things like cotton, rice, alfalfa, and so on. There are lots and lots of genetically modified plants on the market now. About 10 different crop types, 120 different types of seeds are available. It's interesting that genetic modification has earned such a poor reputation in our culture because a lot of the genetic modification that has gone on has allowed us to grow crops with fewer poisons being applied to them. So overall, we use fewer herbicides to kill weeds. We use fewer pesticides to kill insects. We've also managed to create genetically modified plants that improve human health. In fact, one of the most famous genetically modified plant is called golden rice. This is a form of a rice that has had genes inserted to produce beta carotene. Normally, rice is not a good source of beta carotene, and you may wonder why you would want to put beta carotene into rice. Well, the answer is that there are parts of the world where rice is a primary staple in the diet, and where there are very few dietary foods available that contain beta carotene. Asia is a good example of this. Beta carotene is a critical component for the production of vitamin A, and vitamin A is critical to normal eyesight. One of the most common causes of blindness in parts of the world is a deficiency in vitamin A. So golden rice was produced in an effort to prevent blindness around the world. And that's what you're looking at here. This is a bowl of this particular species of rice before it was genetically modified. And you can see it just looks like plain old rice. This is golden rice. This is the rice that has had the beta carotene gene inserted into it. And so it's very rich in vitamin A. And by growing more golden rice around the world, we have managed to improve the levels of vitamin A in people's diets, and we have reduced the amount of blindness that has been a consequence of vitamin A deficiency. So, yes, a lot of people are very concerned about the introduction of genetically modified organisms and genetically modified foods into our culture. But the truth is, there have not been any incidences reported where anyone has become ill or died because of exposure to GMOs. And the truth is that GMOs allow us to make a whole lot of products that improve our lives, including a lot of the products that we really enjoy eating. So yes, genetic modification is here and it's here to stay. And as I said, it is not something to be feared. So bacteria, yeast, and plants are commonly genetically modified. We have also done some genetic modification in animals, but right now there is only one genetically modified animal that's available for consumption on the market. So we have genetically modified sheep, goats, and even cattle, but not for human consumption. A good example of this is a species of goat that has been genetically modified to produce spider silk protein in its milk. So while these goats produce milk naturally, while they lactate, one of the proteins that is produced in the milk is the same protein that spider silk is made out of. This spider silk protein is incredibly strong. In fact, spider silk is one of the strongest textiles, if you will, one of the strongest fibers that is known to man. So there's a lot of research being done on how we could produce spider silk protein, again, through these genetically modified goats, that we could then use commercially. There are lots and lots and lots of laboratory animals, including mice and rats and so on, who have had genetic modifications. One of the biggest genetic modifications that are done to laboratory animals are what are called knockouts. When we want to study a disease, we need an animal model in order to do that. And one of the ways we create animal models is by knocking out a functioning gene in that animal through genetic modification. For example, let's say you were interested in studying the disease cystic fibrosis. Cystic fibrosis is an inherited disease, and it involves a single amino acid being out of place in a protein. You can knock out the gene in these mice for that protein, and you can essentially create a mouse that has cystic fibrosis. It no longer has the normal copy of that gene, just like people with cystic fibrosis no longer have a normal copy of that gene. So knockout mice have been a tremendous advance in our ability to study human diseases because we've been able to create a model in the animal for that human disease. I mentioned that only one genetically modified animal has been approved for human consumption. That approval came in 2015, and that approval came for what are referred to as aqua advantage salmon. These salmon have been modified to contain a gene that allows them to grow at a much faster rate. So what you're seeing at the bottom of this slide is a normal salmon of a particular age, and then the aqua advantage salmon at the same age. You can see that the aqua advantage fish is much bigger than the same fish that has not been genetically modified. Now, the advantage of this is obvious. It allows people who farm salmon to grow those salmon to size sooner. So they spend less time feeding the fish and growing up the fish to their market weight. They can send them to market much quicker and for much less money. Now, the other animals that you can see on this slide, you can see a sheep up here, and you can see over here some genetically modified mice. This one is a breed of mouse that has been created to have a gene knocked out that produces a hormone called leptin. Now, you may have heard of leptin before. It's involved in our ability to know when we've had enough to eat. So the mouse over here on the left has a normal leptin gene, and the mouse over here on the right has had its leptin gene knocked out. So this mouse no longer makes normal leptin, and as you can see, this mouse has become obese. In other words, in the absence of the leptin hormone, this mouse doesn't have the ability to know when it's had enough to eat. And so it overeats and it becomes obese. Leptin has been a big area of research. The physiology of leptin has been a big area of research in the study of obesity.